Welcome back to the DemystifySci podcast. I'm Anastasia.
I'm Shiloh.
And this is the place where we play the game of figuring out what the world 10, 20, 30, 100 years from now is going to look like. We are scientists by training, and at some point in our careers, we realized that there was all of these ideas that had gotten abandoned in the past. So we have all of these consensus perspectives on science, but tons of good ideas are just left behind. Everybody's forgotten about them. People don't have time to study them. And now, as we're running out of steam on a ton of the current paradigms, we've realized that there's a tremendous amount of riches that have been left behind in the archives of the people who came before us. And so today, we are exploring theories of water and biology and biochemistry. This is a follow-up to a conversation that we've started on the show where we've talked to people like Tom Seyfried about the role of metabolism in cancer, Stephanie Seneff about the effects of glyphosate on metabolism, about Barbara Corky on the effects of obesogens in our food supply on various metabolic processes in the body. And today we're talking about what we've previously described as being the dark matter of biology. So we have this friend, Michael Hughes, who is a biochemist who studies water. Shiloh studied water for many, many years, and the three of us together are trying to figure out how to tell this incredibly complex story about what water, electricity, and chemistry are doing together inside of living organisms in order to power the super weird processes of life.
Which might not seem radical, but there's a very standard paradigm that has triumphantly emerged throughout the 20th century that everything in biology flows downstream from genetics. And certainly all of our biological, biomedical technologies center on genetics, and there is a lot of power in being able to decode genetics. But it seems like there's a revolution underway where processes actually feed back onto the genetics in a way that might be at least equally as powerful as the other way around. And that's at the core of what we're talking about today.
There's a concept that kind of covers some of this stuff, which is called terrain theory. And terrain theory got really wildly popular on the fringes during the pandemic because people started to realize and to wake up really for the first time as we dumped millions of dollars into the study of viruses that for whatever reason, not everybody gets sick from a virus. Like even something like Ebola. I remember when we were teaching the immunology of COVID course, I looked into Ebola because I was trying to figure out like the background rate of like what is the death rate of Ebola. And there was this astounding paper where a group had gone to, I don't remember what African country, where there had been an Ebola outbreak, and they realized that like 70 to 80% of people had had Ebola, but none of them had had symptoms. And that was this like totally mind-blowing experience for me because Ebola, it's like it's supposed to make you like bleed out of your eyeballs, 99% death rate and all the papers. And these scientists are going out, they're actually measuring what's happening in the population, and they're realizing that like, oh wow, like different bodies do different things.
Polio is a good example too. I think it's like 1% of people who get infected with polio end up with the paralysis that it's so famous for.
And then do you remember that COVID challenge study?
Oh my God.
Like there was a COVID challenge study, which was, so it was one of the last papers that we taught. It was the second year that we taught the immunology of COVID class. And it was a Nature paper where they infected like 40 healthy young people with COVID. And then they did all these tests and they were like, man, it's really crazy. There's no correspondence between the number of copies that we can detect by PCR and the severity of symptoms. Like if you look at the graph, it's just this like shotgun blast with no correlation. And so crazy things are happening. And Michael Hughes is doing a postdoc at St. Jude's in Memphis right now, where he's trying to piece this entire story together. He comes at it from a biochemical perspective. That's his background. He's like a protein crystallographer. He understands how proteins work, all of their electrical properties, the way that they respond to changes in their electrical environment through like ions and everything else. And now he's trying to see what the picture of how these proteins interact with water says about the functionality of the body and what it means to maintain like a healthy homeostasis where things like Alzheimer's plaques don't form or cancer cells aren't able to propagate or...
So yeah, it's like, it's more of a flow than a homeostasis from what I understand, but we'll get into it in the conversation a bit. Because maintaining a healthy body seems to be about keeping things really far from equilibrium, actually. which is why being a being is extremely strange in the world of physical objects.
Oh, we didn't even get into that, but
A
it's
little bit.
a little bit. I mean, it's a really interesting conversation because we're literally trying to figure it out here in real time. So like Michael has written this massive paper where he's just put everything that he knows about water and osmosis and proteins and disease into one place, and it's an encyclopedia. It just contains every little piece. And so we're trying to figure out what is the story to tell around that? How do you weave all of these different pieces together? How do you understand the behavior of water inside of these crowded systems to let you see something that simplifies biology? Because at the center of biology is metabolism. And metabolism is insane. We talk about this very early, where if you look at the way that we chart metabolism, it's this massively complicated map of inflows and outflows and molecules that are moving around the cell and doing things. And there's no systematic way for being able to look at that and be like, oh, I understand what's happening here. But what Michael's working on is he's working on an ability to say, there's an overarching principle. You can take all of this complexity and you can simplify it.
And since there's endless details about that, this is definitely not the last conversation we'll be having about this. And we love hanging out with Michael anyways. Plus, maybe next year we'll do a conference surrounding some of these issues with water. This year, obviously, conference surrounds cosmology. And if you haven't already got a ticket, you should consider coming out to Portugal and hanging out with us this summer. It is June 12th through the 16th in Sesimbra, Portugal.
Yeah, so basically we're selling memberships to the conference this year as opposed to tickets because we are inviting people to join our scientific society. As a reward for joining the scientific society and funding the project, people who buy in at the stakeholder level, which is now officially sold out, are invited for 4 nights to this beautiful hotel in Portugal. They get to see the talks, they get to the discussions, they get to go to all the dinners, to the opening and the closing ceremonies. And then everybody who buys in at a lower level gets progressively slightly less perks. And so at this point, you can still buy in at the lower level. You can support the project and come to Sesimbra, enjoy all the talks, come to most of the dinners. The only thing that we cannot offer you at this point is lodging. And so we would love to see you there because it looks like it's going to be, I don't know, I think they were probably going to end up with about 50 people there. It's going to be a really tight-knit group. If our last conference was any indication, it's going to be phenomenal. Our listeners are some of the best, most interesting people that I've ever come across. And so when we get them all in a room talking about subjects that they love, it's really electric.
Otherwise, if you can't make it to the conference, remember, guys, we work directly for you, the listener. So if you can shave off the tiniest piece of your paycheck to make sure that we can keep doing this project and put more and more of our time towards it, come over and check out our Patreon page. It's patreon.com/DemystifySci. For as little as 75 cents a week, you can make sure that we can keep the lights on here, that we can travel and do in-person interviews, that we can just be a more powerful platform for these investigations that nobody else is doing.
There's literally nothing that you can buy for 75 cents a week. There's nothing. I'm racking my brains of what you can actually do with 75 cents a week. And you can't even buy a pack of gum with that. But you can, through the power of large numbers, make sure that this program never has sponsors, never sells you any garbage, and is free to pursue the most exquisite scientific inquiry that is available anywhere in the world right now.
And if you're already supporting the project, you know how we feel. Thank you so much.
Absolutely.
We can't really say anything else besides thank you, thank you, thank you, thank you, thank you. Otherwise,
enjoy the conversation with Michael Hughes. Tell us what you think in the comments. Tell us what we should talk about next, what you want to see more of, what you want to see less of, and come to Portugal.
And one last thing. If you have a theory you want to present, consider checking out our new segment called Paradigm Drift, where we give you, the audience, a chance to present your idea that might change the world. So check that out. We'll link it down in the description. It's a once-a-month affair, and the next one will be on May 9th. So I would love to see you there.
Enjoy the conversation. We'll see you next time.
The scientific revolution starts now.
The DemystifySci podcast.
Let us see in the dark.
As I was reading your paper, it occurred to me that what you are proposing is something along the lines of the body being an electric universe and water as this almost the ether of that cosmology.
And we have
Yeah.
hit bingo in the first 30 seconds.
It's a good way to start. And what's so strange about this journey is I was early on a few years ago, I was talking to the postdoc I sit next to, and this person shared, it sounds like you're describing like water as like the space-time as which all of gravity's computed to dictate how things like move around. And so I like independently, I didn't see it then, but then I was like, yeah, that makes so much sense because after going through my water journey and realizing like how water interacts with electricity, I was like, man, I think that there must be an aether that I just that was what showed me, or like I started to research aether theories of physics after the water findings that I came across because it seemed like there must be some kind of mechanism which does this outside of water, right?
I think there are a ton of really interesting parallels between cosmological science and what you're doing. I actually could not help myself as I was reading your paper from thinking about the liquid sun model because so many of the convection argument, you have these convection versus diffusion arguments at some point, and I started thinking about the way that heavy elements would be conveyed to the surface of the sun under Pierre's liquid model, and yeah, it just seemed hard to not think about that stuff.
Yeah. And I think it's so cool because as like thinking about diffusion and things moving at the micro scale, like it turns out convection dominates over just kind of like normal diffusion, right? Convection and so osmosis actually is better modeled as like a large convection component because it's so much faster than just diffusion at the atomic level. Right? And so in this sense, convection represents kind of like I'm not sure if you've ever seen I love this YouTube video, but if you look up caterpillar like there's a species of caterpillars that they'll migrate. So it's a speed of the caterpillar, but then they'll form this ball. And so when the caterpillars, like what they'll do is they'll be caterpillars crawling on the bottom, but then the ones at the end of the train, they crawl along the top to go become the front of it. It turns out this caterpillar ball moves faster than the fastest speed of the caterpillars that compose them because you get to have your speed plus the underlying caterpillar speed when you're going to the front, right? So they all move together more quickly. And in this way, I think this is kind of like an intuitive idea of like what convection represents. It's this like net flow, and then diffusion happens like is the individual caterpillars within that pile. But convection is the whole pile going in one direction. And so, of course, looking at the liquid sun model and seeing these huge like blobs, these convective plumes, or even like your pot boiling, recognizing that in the cell, in the test tube, we kind of go to like a thermodynamic equilibrium. But the cell is by definition kept away from thermodynamic equilibrium, which invites in these huge convection currents. And it's like a totally wild way, but it kind of turns the cell into like this energetic vortex, right? Where what really matters is like everything moving together is faster than looking at the individual components in a test tube.
Well, and it's really interesting because the paradigm for biology as of right now is kind of this receptor ligand paradigm. Like, yeah, you have DNA. DNA stores information, and the way that it transmits it is through proteins and signaling molecules, and everything is the dance of these macromolecules. And more, I think we're starting to realize that that's kind of incomplete. Like Michael Levin's work is suggesting pretty heavily that that's incomplete, right? Because he's basically showing that, and you mentioned this, that there's something bioelectrical that you can modify, which creates these permanent form factor changes without changing the DNA. And so we know that there's something that's not really encompassed in the current models of biology. And furthermore, like if you look at the foundation of cells, it's metabolism, right?
Right.
And then everybody who's ever looked into metabolism has probably seen the like Roche Pharmaceuticals metabolism map, which is literally a like 8 by 12 foot poster that is just covered in 10-point font of all of the different stuff that's inside of metabolism, all of the different flows.
Like signaling pathways and kinases and everything.
And like NAD and like all of these redox active molecules. And so there's this huge interplay. And I'm like, this feels insane because you can't figure it out. There's no overarching organizing principle.
I actually took one of those from one of the first conferences I ever went to when I was just a little baby scientist. And I put it on my wall and I was just like, oh, maybe if I just stare at this long enough, like some brilliant insight will pop out at me. But I think it was like you're saying, it was almost just this shotgun of information that had no obvious relationship.
Well, and I think that this is how scientific progress happens, which is that first you have to make the map, right? You have to figure out like what pathways feed into what, how these things are linked. And then after you've done that, you have to do this next level of understanding, which, so you sent us this, 80-page paper, six different sections. And I thought what was most interesting is that the pieces of it that felt most actionable were the biochemistry, where you were looking and you were like, look, this is the charge on this molecule. This is the size of this molecule. This is the electronegativity of this molecule versus that molecule. And so if we start to zoom out and instead of just preoccupying ourselves with the sheer complexity of the pathways, we can start to tease out rules for how these things operate that let us actually understand a higher level picture that feels simpler somehow.
Yeah. And thank you for like highlighting that, bring that. I think that's what I'm so like in a sense like desperate to get off my chest is I experienced that same thing of looking at that Roche poster and like in the hallways of my undergrad and just like walking by it and then being like as you mentioned, Shiloh, like can I like get a little bit more today than I did yesterday? And the answer is like no, right? And so what's strange is uh like the periodic table, I love. I used to have a chart of like the nucleotides. I mean, it's this huge, incredible amounts of information, but if you learn how to read it, it's just very intuitive. And the metabolism poster isn't that. So what this journey was for my particular like science career is like as COVID happened, I started to like try and research what is underlying this like phase separation field that I was looking at. And I come across this huge osmosis and electric component and all of the electronegativity. Like there are some underlying rules that I found that was dictating everything. And so suddenly, over a few years, this all went from incomprehensible to there's this few like paths that are rail trod, and everything else is kind of like branching out from that. And I think that just helped me so reorganize what biology and what biochemistry is. Right, there's very specific tracks that the cell wants to be on, and we can understand that language now.
And physics too, right? Like it almost seems like you've discovered that surfaces and the distinction between different objects in the cell, let's say, come down to this phase like interface, like interfacial chemistry, essentially, right? It's almost like these
Absolutely.
physical bodies are defined by these interfaces.
In fact, I would argue that you know, I'd like to borrow this kind of from like Michael Levin's work again, but he's now been on this kind of like computer science bent of talking about computer science algorithms and how this shows up in biology. And there's so much insight that we learn from that. But one of his that he shared is that the most compressed information looks random, which means as you compress a file and you turn your color image into just a few colors and then the XY coordinates which they live, it gets smaller and smaller and it looks more and more random. And then you just like unpack it with the correct algorithm for a PNG or JPEG or something. And so your cell does this with DNA, right? DNA is this huge message that's so condensed and so compressed and it looks random, but then your cell unpacks it and it impacts it into the cytoskeleton, to all this signaling, right? You just turn into all these. So you recreate this huge repetitive structure from this really condensed random information. What it does is it maximizes interface, right? And if life happens at the interface is represent physically what it represents is an electrochemical gradient or an electrical gradient. And this allows catalysis. This allows so many things we use electrical gradient. So anytime you find an interface, it's almost as if life is like unpacking the cell's information to maximize a biochemical interface.
That's really interesting. Are we
And
gonna
in
say?
fact, like
Yeah,
this is a
can you
capacitor.
can we define interfaces as, do interfaces necessarily have an electrochemical gradient? I guess they could be at equilibrium, but inside of the cell.
Okay, so intuitively, an interface technically is. Does it have to be in equilibrium?
No, I think it's not in
No,
equilibrium is the trick,
Yeah.
actually. Inside of the cell, anyways.
in my test tube it's at equilibrium, but in the cell it's kept out of equilibrium. And when the cell reaches equilibrium, it's dead, right?
Oh, that was what was really interesting too. You had this line in your paper about how the difference between in vitro and in vivo was that in vivo systems, they're always extremely far from equilibrium.
That's right. And that's how you induce mass to travel through the cell for metabolism. Because when you hit equilibrium, there's no more osmosis left. It's just diffusion next to your neighbor, right? Uh, and so That's what's so important about like the metabolism is that the metabolism is the flow of like information through the cell. And when you don't have metabolism, you don't have a difference to draw new nutrients in or put something out, then yeah, you like end this. And that's actually what all my test tubes look like is like the end result, the dead cell, right? But the actual living cell is always keeping an imbalance. And this is what draws or induces mass transport, which is metabolism. Metabolism is trying to draw your glucose in any pieces you need. And then to get something in, you have to give something up. Something has to go as well, right?
Okay, so let's see if we can zoom out a little bit and make a model of some kind for life. Is that too ambitious?
I maybe I'll struggle here, but I think that there's one thing that comes to mind when you mention this like model for life is this like low entropy kind of like resting state, right? This like ordered resting state. So what I said before is you unpack the DNA into a bunch of protein structures, and these protein structures make a huge interface. Um,
But you have to continuously have stuff that flows across the interface, right? And so there's something in the work that you've been doing that seems like it is all about the maintenance of an interface that's able to pull the things that the cell needs into the cell and get rid of the
Correct.
things that it doesn't want anymore.
Right, because when you don't do this correctly, this becomes cancer or neurodegenerative diseases, kind of like a sickness manifestation. So, what's cool about like how you draw things in or how you get them to go away is that the water is what's mediating this computation, right? So you draw in. So when we're talking about glutamine, glutamate, and this is maybe a reference back to a previous podcast with Dr. Seafree, but glutamine and glutamate are two amino acids that your body needs for metabolism. And the difference between them is that one is kind of got like a protein side chain that's neutral, that's glutamine, and then glutamate is charged. It's salty. It's actually got a negative charge on it. So when you take in glutamine and you do work on this in the cell to turn to glutamate, now anytime your body does chemical work on like a substrate, which means just like eating glucose or eating glutamine and turning into something else, the end product has a different relationship with water, right? So now things aren't just randomly diffusing around the cell. Glutamine wants to travel to the liquid environment it prefers, which would be in the cell where the metabolism happens. And then glutamate, this other thing, wants to travel to a different environment where it's ready to be a signaling molecule or go do some action. And the reason why the glutamate will move to its new environment is because the negative charge changes its hydration environment, changes how it wants to move from one interface to another, right? So your interface sets up these large interfaces, and then the small molecules metabolism and all the proteins that are signaling, they travel between these interfaces from one part to another part. And this is the trafficking that leads to life, right? Is that as you modify the chemical substrate, it needs to find a new home where it's happy at equilibrium. And this is how we keep inducing this like cycle of metabolism.
Okay, so bring phase into this because phase is a huge part of what you're doing in terms of this like did you coin the term liquid phase separation or is that well known?
That's relatively well known. What's kind of funny about this is how my like PhD started in 2012. Was I joined a lab studying amyloid diseases, and so that includes a phase transition where you have a protein that's an amyloid protein. So like A beta in Alzheimer's disease, and it's happily floating around your system, but at some point in your life, it starts to phase transition into a solid amyloid fiber. And what the field started to realize around like 2015 was that there's another stop along the way where the amyloid proteins, they tend to go to this liquid phase separated droplet, and then it can subsequently become an amyloid fiber after the fact. And this liquid droplet is what became so interesting to us because polymer scientists had seen this forever, but bioscientists hadn't quite appreciated that this is happening all the time in the cell. And in fact, I read a really wild 2006 review by a physicist who said we should see liquid phase separation in the cell all the time. And because we don't, it tells us these things about the cell. And then 10 years later, every one of those reasons he lists, we see like the cell actually does this in metabolism or violates it. So a phase in this context, liquid phase separation is where if I have my amyloid protein, I can just change the temperature of the test tube, and it suddenly goes from being dispersed and monomeric in solution to forming this dense liquid granule. And this dense liquid granule, it's not amyloid fiber, it's not the solid phase. They still can diffuse over each other, but it's spontaneously concentrating these proteins in this one aqueous environment, almost like oil and vinegar separating for your bread, but it's supposed to being oil and water. There's water inside both. So what is it that causes the protein to concentrate together and like fight entropy? Because entropy tells us it should fall apart, but instead it's just spontaneously ordering. So what is really cool about this for biology and the revolution is that it gives the cell a way to order its macromolecules, order where the proteins are going, where the solutes, where the glutamine, glutamate wants to go, all without wrapping it in membranes and using transporters to pump it across the membrane. Instead, you
Just
can
this
just
is just
rely.
by preferential orientation of it towards water molecules, essentially.
So this each molecule will construct its own water shell around it, right? And so when you're diffusing and your two biomolecules diffusing in water, you don't see the other biomolecule. You first encounter its hydration shell, and that hydration shell is associated some structure associated with it. Not that the water is like necessarily like stuck in the structure to say, but they're compatible and incompatible hydration shells. So
Mm.
this is what. So before you even touch each other, you first coming across this water shell and either jives with what your water shell is as the macromolecule you are, or it doesn't. And this will lead to spontaneous phase separation of leading to that ordering that I was talking about.
So it's kind of like a first order blueprint for where the different pieces should combine.
So this is what it's becoming. As it turns out that a lot of like the signaling that we see in cell is these phase separated droplets in the cytoplasm. Where if you see two signaling molecules like interact in like a bright puncta in light microscopy, if you look into it, this is going to be obeying the physics of liquid-liquid phase separation, and that's what it means. Is these cells find their way and stay together. They don't diffuse away because it's more energetically favorable to hang out within your own preferred hydration environment than outside.
And so, like, you talk about this pretty extensively in the first part of the paper, where different types of you can mix polymers in solution and then end up getting like two different phases where one polymer associates primarily with itself, the other polymer associates primarily with itself. And so, technically, it's like a bulk environment, but there's structure inside of the bulk environment that emerges spontaneously. This is kind of what you're pointing to here.
I This is one level looking at your cytoplasm does this because you spontaneously make like microtubules, right? And that's of course like kind of a liquid to a solid phase, right? Like a microtubule is a more solid, like a cytoskeleton component. But the thing is that they'll spontaneously happen and it obeys the same kind of rules where in order to like minimize the energy of the system, it tends to actually lead to the formation of ordered like this like low entropy ordered structures, and this is a strange cost of minimizing the systems, the free energy of the system, is that you do get spontaneous, like low entropy ordered states. And this is what these liquid phase separations like represent. Uh, and so, uh, yeah, I think that's how I like go for that. But maybe I didn't quite get what you're going at with there.
No, I think that covers it. I mean, I think it's there's so many ideas here that it's hard to get a single thread that weaves through all of them. And so I want maybe sticking with something like amyloid proteins as an example is useful. So amyloid proteins are I know so little about this. Are they produced inside of cells? Are they in the interstitial spaces? Like how does can you give us some background on that?
Yeah, so the original way that we kind of come across amyloid proteins are from like Alzheimer's disease, and these some of these like neurodegenerative diseases identified 100 years ago. So in the cadavers, the early scientists would open up, do some staining, and see these fibrous that these stains would stain these like fibrous inclusions, and they were originally called amyloid because amylose, like sugar. They thought that it was sugar, so it's like misnamed. But then we come along and find out that they're proteins. And so for each different disease, Parkinson's disease, ALS, Alzheimer's disease, there's usually some different proteins that are involved in forming these amyloid fibers. And we usually associate them with things going like wrong in the body. And severe symptoms coming from that. But what's really curious is that as I was going through school, is how we were starting to recognize how prevalent amyloid is throughout all of like biology as a useful structuring motif. So biofilms, they're made from amyloid. So bacteria will excrete protein, and outside in the bacteria, totally harmless. It just is freely diffusing. But outside the chemical environment, it's different. Suddenly, it forms an amyloid fiber, right? So
And this is like
Amyloid.
a really persistent structure, right? So if you know anything about biofilms, you probably know that they're really hard to get rid of. And the reason they're hard to get rid of is because it's this really thick, gooey outer shell that basically the bacteria exist inside of, that's very hard. Like antibiotics don't diffuse through it properly. It's very difficult to break up even with stuff like bleach. Like it's just a really
This
Right.
is why, like, fixing what like what do they call it? Uh, irritable bowel syndrome or something like this, right? Like fixing a gut that's gotten out of order is really, really hard because you have to essentially tear up these impossible biofilms.
exactly, yeah.
Right.
And so amyloids are central for that. And so inside of a human, what is the normal function for amyloid?
So there's a few answers to this because when I first started kind of doing my research, it was under the guise that like amyloid is bad, and but now what's changed and become aware is like your body actually does harness amyloid for some normal functions. So in Alzheimer's disease, some like bad ones is that amyloid beta plaques in the brain. They're formed from an extracellular fragment. Tau is another protein that forms amyloid fibers. It's an intracellular, it's a microtubule binder and stabilizer. So it can be outside, it can be inside. It just kind of depends on the situation. So so amyloid is this really general biological phenomenon, right? And so what does it represent? It kind of represents again a state of matter change, a phase transition from being liquid to a solid, and you can actually use this in your body for helpful things because uh, you can store things like insulin or proinsulin or PMEL is a skin pigment and is stored in these granules that when excreted into the blood that can diffuse, it can actually like dissolve. So in some ways, your body uses the amyloid fold to store and sequester some molecules it wants later, which will then dissolve nicely when they enter the new chemical environment like the blood. Right. We actually have evidence of your body having healthy amyloid and useful, and what was strange was for my PhD, I was working on these ALS proteins and I was trying to find the disease related amyloid for them. But this paper came out that was talking about how the amyloid fold that these proteins form are there, it's really weak as opposed to this really strong structure that Anastasia had mentioned. It's about as strong as like a copper wire by thickness and really hard for your body to get rid of. You can't autoclave it, right? You can't do all these things. Turns out these amyloid fibers that were forming from these ALS proteins were really easy to melt by like a subtle temperature change. And the theory that emerged was that this might be actually useful for gene regulation because you can kind of get the gene regulators you want together in this amyloid fiber. And as opposed to, and that might be how your cell like actually organizes gene transfer. This may not be the case now. There's some evidence that's like still in support of this, but I tend to go by this turned into the liquid liquid phase separation model. Is that now you're not actually using, it seems that that amyloid port of those ALS proteins makes the liquid environment to allow liquid liquid phase separation, which then recruits the other genetic factors like from around. And if not, like recruit seems weird because it sounds active. What happens is if you're the other factor, you diffuse in and then it really sucks to leave. So you stay there, right? It's not like a recruiting per se, but that's kind of like a biophysical version of what this looks like. So it really changed what amyloid proteins were meant for me and how I viewed them and what I would like do with them later, right? Is that your cell seems to be harnessing this.
And so, does that give any kind of insight to treatment approaches? Because if it's something that is normally in the cell and functions properly, because ALS is supposed to be a genetic disease, and so is there something about the protein
What
that's
you say supposed to, like you're not buying it.
well, I'm just saying that if the problem is some kind of misfolding that accumulates over the course of a lifetime, is it in an environment? Is it a mixture of environmental and genetics where there's some kind of slightly malformed version of the protein that's more likely to clump that can be somehow mitigated through trying to modulate the external environment, the interstitial fluid
Right.
environment? Because I
So
don't, at the last paradigm drift, somebody was talking about like alkaline therapies
yeah,
for cancers, and I was like starting to think about that in terms of things like amyloid plaques, because if they're forming on the outside of cells, homeostasis generally is something that we think about as being inside of cells, and then there is definitely some homeostasis just generally in the body, but it seems like it would be easier to mess with that homeostasis than to really regulate the interior cell environment.
Yeah, absolutely. So I think that that's kind of like the journey that I had was our original way to view uh, amyloid disease progression was this thing called the amyloid cascade hypothesis, and this is really about crystal seeding, right? If you put if I have a super saturated solution, really like hot water filled with tons of sugar, and then I drop a one crystal of sugar into it after it cools, it'll just drop out all the rest of that. So our original idea was that your cell is so concentrated with protein that it always exists in kind of this like super saturated state. And we're worried that maybe this like very correct seed crystal comes along, and this is actually like mad cow or Creutzfeldt-Jakob disease. Is that you eat this like diseased meat with the correct protein prion, and then it gets in your body and starts to seed itself template from your own body's proteins and build out, and this leads to that disease. So this was the paradigm from which I entered, and in 2017 or 2016, right, there's like a paper in Nature about an FDA approved drug for Alzheimer's disease, which actually uses your immune system to get rid of the fibers. So this drug can get your immune system to start eating them. So the consequence is that you get rid of the fibers, but the symptoms don't change. You still have all the disease. So uh, the seeding model now is like weird for the amyloid cascade hypothesis because even with getting rid of the amyloid fibers themselves, we're not fixing it. So I, as a protein crystallographer and someone who's studying like structure and doing all this, I love the structure, right? Like of you, but it seemed like I was missing something, and this became apparent with it seems that what controls whether you form that crystal in the first place, whether you do that phase transition, is as we've been discussing the like aqueous environment, the liquid around you, with the water, the pH, and how concentrated other things are around you. And what's crazy is during this paper, I was like trying to search for things that seemed like it would support this, and I came up with that. And when they sampled patients' CSF from Alzheimer, from neurodegenerative diseases, that they found that it was a bit more acidic than controls, right? And so suddenly, this is works so much better. Is that you now have changed your extracellular environment to the incorrect aqueous solution to keep these things happy, these proteins nice and soluble. Your disease condition looks like an acidification of the CSF, which then would induce the aggregation of A beta and lead to this disease. So it's not even the protein itself that's a consequence, potentially a symptom. What the real thing we're looking at is the change in acidification, and what this represents is a change in the transport of molecules in and out of the CSF, right? As I've been kind of like alluding to. So this suddenly turns a rare protein seeding phenomena into a metabolism issue. How do you mass transport in and out of the CSF?
Because you
That kind of
define
issue.
metabolism as some sort of switch on mass transport. Is that correct?
So I see metabolism as the flow of like physical mass through the cell, right? You're going to need to take in food and then excrete waste. And when you stop doing this, and so you're going to need to take in something that's going to like run down an energetic slide, and you extract a little bit of energy from that, right? But then when you stop having this thing able to run down its energetic slide, you can't extract energy, you're not doing metabolism, you're now dying, right? So it's really, really important to be able to have the correct conditions for nutrients like want to travel from one phase of the liquid in the body to the next phase of liquid in the body and so on to do the proper chemical reactions to like extract that, right? And so then in this process of extracting some energy from this, you actually get to harness that for, again, like the mass transit is kind of tied to the mass moving through. The water is perfectly reflecting uh, the water structure changes based upon what you dissolve in it. And so as you change what's dissolved in it, it like drags that water with it. So when we're talking about osmotic flow of things through the cell, it really is dependent upon the modifications that you do to these small molecules to extract energy from, and then how that brings the water with it also.
So before we dive deep into like osmosis and what that really means, I want to ask some questions about like the physical state of the body. But what were you gonna say?
I was just going to comment on it. Feels like we are in the birth of a soil over seed revolution in biology in general.
Sounds really
Yes,
nationalist.
I
like
know, I
it.
know it does. Well, it kind of like corresponds to like the Maha movement and stuff, as far as I can tell, right? It's this, you know, people were guaranteed the miracles of modern medicine, which is largely a seed sort of approach to reality or towards medical reality, health, whatever you want to call it. And that's in some senses played out to the best of its ability, I feel like, and people have started to find success through other means, largely trying to keep the soil healthy for the life form to exist in.
Well, this is terrain, right?
Yeah, exactly. So it's almost this Darwinian pushback, right? Where, terrain theorists are so stupid, haha, and then it's kind of leaning back the other direction now. I feel like this is something that needs to be negotiated in terms of approaches to biology, and it feels revolutionary.
And I feel like this is exactly like what Michael Levin is getting at, right? Like the genetic expression of the cell is not due to the genes, it's due to the environment around it that like draws that out of the cell, right? And so what we're really getting at, or what I'm trying to pose, is that this osmotic flow, this metabolism, there's direct biophysical consequence for how the cell will express its DNA due to these flows and information transfer. And this is nice because it kind of like adds the mechanism to this, but what it really is reflecting exactly like what Michael Levin is kind of getting at, that floors the whole biology world, is that your genes don't tell you who you are, right? The soil tells you like what they get out of the genes in
Well,
this
his
like
work
metaphor.
on galls is so valuable for this, right? I mean, he hasn't done a lot of experiments on galls, but so I don't know if you've seen these before.
Sure. I'm
So
not sure if I
if
have.
you probably have when you go into the woods. Have you ever seen like an oak tree that has these weird lumps growing on it?
Okay. Yeah, yeah. I think
And
I know
it's
what you're
these
getting at now.
parasitic wasps that lay their eggs inside of the tree and they grow these nodules. And the eggs mature in the nodules, they eat the nodules, and then they leave and the life cycle continues. The gall is not a genetically distinct structure. The wasp did not make that gall. The wasp shows up and it does something to the tree to induce it to grow a completely different morphological structure. And the diversity and variety of what insect galls look like is astounding. It's like a normal leaf can grow this insane filigreed structure that's like bright red and looks nothing like a leaf. And so there's this entire space of biological manipulation that seems so obvious when you see it. Like this is my favorite kind of science where you're like, well, yeah, like obviously it must work that way when you look at it because it's happening in the world already. And so the terrain theory was really maligned because it was the antithesis to germ theory. And so
Okay.
in the like late 1800s, there was this guy, Virchow, I think his name was, and he was the antithesis to Pasteur and the germ theorists. And so the germ theorists were like, disease is caused by germs. These are the principles of how we can figure out if disease is caused by them, which is that you can find some kind of microorganism that you can isolate that can live outside of the body that you can then transplant into a healthy organism, and then that healthy organism succumbs to the disease. And that's been the paradigm that we've been working on, that it's like the soil doesn't totally matter
It's like a
because
completely polarizing argument every single time, right? And there's this backlash. So, of course, the germ theorists went out, but, there's some like caveats and nuance to it, right? Because before the germ theorists went out, they were the idiots in the room, right? Everybody was like, no, it doesn't matter at all. And now it's kind of swung back in the opposite direction.
I think that we're living through an era of synthesis where
I
it's
agree.
like there was this period of science where there were these multitude of ideas, and then one idea wins because for whatever reason it's more popular, and we run with it for as long as we
Or
can.
even
Like
more immediately productive.
exactly, yes. It's like you have a paradigm that you can mine for some kind of useful products, if that's medicines or that's, like figuring out the genetic cause of disease. And then you get to the other side of it and you're like, okay, well, the vein is done. Like we have tapped this out. Where to now? And so now we have to go back and we have to say, okay, well, where were we 100 years ago? Where were we 150 years ago? What ideas
This is almost
lost?
an archetypical experience
So
in science.
amazing.
It's so
It
fascinating. It
is.
seems to play out over the course of centuries or longer sometimes.
Mm.
Yeah, it's interesting to be, listening to this podcast and seeing other people's versions of this and historic other versions of this and go through and how, like, uh, I was not a philosopher or anything growing up, but I thought it was, kind of like goofy, like Plato's like ideas, kind of like this perfect idea, and then its weird incarnation in the world. But now suddenly, like back to that being like really interesting, right? And I think that is exactly what you were saying of we adopt these paradigms, we mind them for as much as it's useful, and at a certain point, we adopt a new one just because it's more useful. Like we ran out of like stuff to do. So I tend to see this as like natural, right? And in fact, like even humans ignoring this cycle seems to be very natural, right? And so I won't like be upset with them for that. It's a going rate, right? I think what is interesting though is, as you say, this age of synthesis. This is where we're at, and this is what's so such a struggle, right? Like uh, I don't really know an experiment to do tomorrow that tells me something because there's so much out there. I know that people have done so much. Like it seems hard to synthesize this. If I were to do an experiment that's based on humans' collective knowledge and just push it that little farther, it's something that we really haven't done yet. It's so hard because I'm realizing is like there's all these different views and paradigms of which I got to like mine so much interesting information, right? One of my favorites was Gilbert Ling, right? I read this guy is a totally different conception of how the cell is built away from membranes and into like aqueous phases. And what's inevitably going to happen is that Ling actually argues that the membrane like may not even exist. Some people in Ling's camp argue that the membrane is not like actually there or like a leak-proof system. And I don't care to like adopt that or fight it, but what they're saying is really, really interesting is eventually if all this liquid phase separation comes around, what's going to happen is someone's going to come back around, realize how important the membrane is, and then refocus it back, right? So how do we is that natural, or is there something that's interesting right now with the internet and synthesis where I think that our job is to just learn as many different paradigms as possible and be able to speak about them?
Pardon the interruption. I do need to remind you that this show is entirely paid for by listeners like you. So if you want to make sure that we can continue to produce this show and do a better and better job at it, be able to create more high quality interviews, do them in person, and basically be able to keep doing this thing, consider coming over to patreon.com and checking out how you could support us for as little as 75 cents a week. It seems like almost nothing, but the truth of the matter is that all those 75-cent donations are adding up to the point that we can basically attend to this project full time, and that's what's necessary to make it the most powerful project possible. So see you over there, patreon.com/DemystifySci.
I think that's kind of like what's incumbent upon our like generation of scientists, like being the jack of all trades, right? Like I thought that as I was going through this journey that, all right, I need to understand water to understand what's going on with amyloid disease, what's going on with metabolism. So maybe I'll reach out to some professors and try and like ask them what's going on, but they have an entire different language, right? They're physicists, they're physical biochemists, they're geneticists, they're not a biochemist with a amyloid background. How do I talk to these people? It became so hard that I stopped talking to them and I just started to just learn it myself, right? Like it didn't save me because if I could even get a conversation, a lot of times the people expect you to use their language
Mm.
in that environment. But we're all talking about the same thing, but with our each of our own fields, like bias of language. Uh, and so this became an issue where I just had to learn it all myself, right? And like in learning it, I'm always like reframing what I'm learning from another field and putting it into my own words from my own field. But I get really good at this game of these people say one thing, they look at it this way, but it's the same thing in this other field and they look at it like a different way. And my favorite and like strangest is like frankly physicists. The physics language is so curious to me. Like I was going to the APS conference where I like met Shiloh as well too, was to hear physicists talk about nonlinear systems. And uh, in biochemistry, and they're ecstatic because nonlinear systems are how you beat everything, right? Like a linear system is that as you add something in front of a light source, so the more that you add in front of it, the dimmer the light source gets, right? In a linear relationship. A nonlinear system would be like when you add like the very first pane, something really weird happens, right? And then as you so at the interface, there's something really weird nonlinear, and it seemed like physicists talked a lot about nonlinear systems because you can play weird games there. You can get out like more than you put in a sense, if linear is always getting out what you put in. And so, but in biochemistry, the first thing that you learn, one of the only things that's really like taught standard is enzyme kinetics. And enzyme kinetics, you get a sigmoidal curve. It's non-linear, like by definition, and you can like play some games to make it linear. And this tells you information. But the first thing that I'm introduced to is like a weird nonlinear cooperative system as the bar for entry. And so it's almost as if at the APS, I was seeing all these physicists come to alive, realizing that biological systems are highly nonlinear. And I just lived there my whole life, right? So again, we're using like the same language, we're kind of looking at the same things, but with such wild synthesis of what it means to us. And I think now it's time to like share these stories between each other, as opposed to like stay siloed, right? And I think that there's so much to be mined from that, but it's a rare job to like come across.
Mm.
What do you think that the route looks like towards the widespread adoption of this new paradigm?
Hold on, before we go there, I feel like that's going to send us in a different direction. Can we close on some of this terrain theory stuff?
That's what I'm trying to understand too.
Okay.
Like regarding it, I mean, there's this age-old story now for everybody who's coming up through the academic pipeline that genes come first and everything's downstream of them.
Yeah.
You know, and like, so I'm more familiar with this at the moment in terms of like cosmology, right? So there's also an age-old story that there's no mediator of light and gravity. It's just bent space-time or something. And I think there's olive branches available in that discussion with people because you can be like, well, actually, if you look at Einstein, he's not trying to totally get rid of these mediators. He's just like, it doesn't matter for these calculations. And so there's kind of this in where you can let people keep it. Another good example would be with the liquid sun that we already talked about. Pierre's always talking about how the sun's not a gaseous plasma, but technically the liquid metallic hydrogen in the liquid form, the liquid metallic hydrogen is a plasma. And so giving people that word plasma and letting them have it is a nice olive branch for that paradigm shift. And I'm wondering if there's one for this shift away from genetics first, that you can let people have their genetics, but somehow open their minds up to this complementary way of looking at things.
Absolutely. So a few thoughts on this, and I think about this quite a bit, and I'm always open to like people's thoughts on it. But I think part of the genetic revolution is that through my career, it's gotten just easier and easier to sequence DNA. So it's a really, really like we have the huge DNA sequencers, right? You can just put you can get single cell transcripts. The power that's available is absurd. So we adopt this tool because we can do something with it, right? It's not because it's like our best or our favorite. It's just usable and there. So I think it's like available. So now we like and using it, we kind of like enter in with this idea that your genes are who you are, and that like poems out there. But now between Michael Levin, this terrain idea, it seems that your environment tells you how the genes are expressed, and this is like kind of cool because like Charles asking like how do people adopt this view that it's really like your gene expression is a reflection of what you're doing, what the terrain is. And so what's cool about this is if maybe it can be adopted if people start to use this as a tool, saying that your diet affects your gene expression, the correct balance of pH can affect disease. If we can start to prove this stuff, like Dr. Seyfried's podcast with you starts to like when people start to adopt this idea of metabolism, mass transport, hiccups in mass transport in the body as disease. And then you can actually start to like unravel this thing that's really hard genetically to understand, right? And so as opposed to genes being telling you what's happening, genes are actually the readout of the biophysical situation, right? The genes are telling you what the terrain is. And so in this way, we don't have to get rid of genetics. Don't stop sequencing and change what you think that the sequencing tells you. The sequencing is the response of the change in osmotic pressure, the change in pH, the change in maybe a signaling molecule, right? And so you can still use it, you just change how you frame it and what it tells you about the environment. And so how do we get people to pick this up? I think that like there's a standard answer here that's like capitalism will fix it, like what's effective will work. And I think that like it's fair, but I think it's also a little bit like dishonest in the sense of like when I share people like my ideas with people tend to be a little bit skeptical, like that's cool. But I'm like, in my mind, I just told you how the world works. It's changed my life. I wake up every day differently because of what I've learned than I did before. But for them, they just heard a random guy like talking to them for 5 minutes after a seminar, right? And so the thing is that if you're actually going to like want to put this information to practice, so you have to let it like change you or interact with you have to interact with it. I'm figuring this out. Thank you guys so much for reading my paper because the feedback that you give helps me figure it out. It is not figured out, right? So a lot of times people talk to me like, "What are you trying to say? What is actually figured out?" I'm saying, I have no idea. I'm trying to get that right. Like I want to know what this means, but this is a dialogue. So there's two ways to get people on board. One, to show it's effective, but more importantly, we have to change how we have dialogues about this. We have to develop a society where there's an interest in changing what you do based upon what you learn. Because around me, most of these people are employed to like sequence this and do this, right? They're not actually employed. They don't get paychecks to transcend, scientific inquiry. They're getting paid to do something else a little bit different. This is really, really important, right? Like I'm currently not teaching grad students. I'm not carrying on the academic tradition. I'm Michael, probably like driving off into some random direction, right? This is an important part of the ecosystem, but it's not the whole ecosystem. So to make the ecosystem available to hear new information, you have to give people like time and space to be able to deal with that, right? And so we expect our scientists to listen to this stuff and to change their ways, but we built a system that doesn't let them do that, right? And so there's one thing that's like a practical aspect of like this information should work and should make you have better health decisions, but then there's another thing to get people to adopt it, which is a change in the social structure or the environment of which we work that would allow them to more easily interface with these
Well, I think
ideas.
as discoverers, it's our job to minimize the impact of what we're saying on people, right? Because inside of these academic traditions, inside of the scientific tradition, people are very comfortable with small adjustments. In fact, they expect small adjustments to their paradigm as the years wear on, right? Like, oh, now there's these, like this junk DNA. It's not just junk, it actually does stuff and modifies, but it's still within this genetic framework that everybody's comfortable with. So, like, that's a small paradigm that really just embellishes the previous paradigm, or at least feels like it does. It might be completely revolutionary, but it feels kind of comfy still to the former paradigm. So I feel like it's our job to make the new paradigm seem like a tiny little leap as opposed to this massive, or like, obviously it's our intuitive response is to be like, this is incredible, y'all should, this changes everything. But that's kind of the worst thing you can do when you're talking to somebody who's comfortable with just everything being kind of solid and moving forward an inch at a time.
Yeah, I feel the same way, and I don't really like know or care to like crack that one. I think that's what's kind of interesting is that it's frankly like a little bit antisocial to just like walk into someone else's like house and tell them how to run the place, right? And so that's not what I'm trying to do, right? People should adopt this because they see it's like powerful. And what's cool about this is like I think the paper I've written in the like line outline that I've like done is like very chemically intuitive. If you have decent chemistry, you can pick it up, and it takes no math, right? I can give you these tools to understand how things like to move and where they like to be without any like math to it. So in trying, like what I'm stuck on is like, how do I onboard these people to these ideas? And that's kind of what this paper represents. Because frankly, if you just open up Gilbert Ling and start reading, it's a horrible slog. It's really difficult. Like I only did it because it was I was desperate and looking for salvation. Right? It's like the last move on the list of things to do. So maybe I can save someone else this like really hard entry like barrier. And so in my sense, this is how I help move it along. Is I just like make it more accessible with like what we already have. Uh,
Well, and
but
there's also this next level of, because, I was reading through the paper, and something that struck me was how difficult it was to get in vivo data. Like, even so, in vitro is stuff that's in test tubes, in vivo is things that's in living systems. And the problem with testing these models inside of living systems is that we're talking about things that we don't really have very good tools to image yet. And so we have kind of there's some really cool things that you mentioned. So somebody came up with an electrode that has a fluorescent readout on it, so you could like, it's very small and you can stick it into different parts of the cell without disrupting the cell. So as it behaves, the fluorescence changes on the basis of pH. And I'm like, that is some astonishingly space age stuff.
Absolutely, right? They are really playing at the edge of material science, computer like acquisition, software stuff to like manage the noise from the signal. It's extraordinary, and I love this, right? Because this is what's changed, right? This is where we have purchased to re-see the world. Is our material science has carried us to where we can ask different experimental questions, even our microscopes, right? uh, I'm trying to use as much as I can a hyperspectral microscope, kind of like how James Webb or Hubble will, for each picture, it'll capture the spectrum, right? So you can look at that one pixel that's a star and see the spectrum of light that made up that one pixel, right? I can do this in a cell with a camera now. So every picture, every pixel of the cell, I have the spectrum of light behind it, right? This changes how we can do science, but it's really, really hard to most other scientists come in with like the experiment, doing the same experiments that they've been taught and carried on for a while. Like, how do you envision a new kind of experiment like these people who made this crazy electrode from bits and pieces? That's really creative. And I think you're right that another thing is like, how do we monitor the chemistry in the body? I really wonder about this because every time you try and measure it, you usually destroy it. And this is the same kind of like quantum like observation issue. Traditionally, when you put a big electrode into a cell to measure a nerve impulse, it's only good for a little bit because the actual like hole that you make in the membrane is so large that it starts to like disrupt the environment around it, and eventually you lose the ability to like get good data because you literally damage the cell. And this like 200 nanometer size or like micron wide needle that this electrode uses, it just doesn't have the same problems.
And
Um,
I'm like always wondering though, from the level of, bodies versus cells, because we know from something like cancer research is what we know is that if you treat something in a dish, it doesn't translate to mice. And if you can fix it in a mouse, it doesn't translate to humans. And so the pipeline,
Occasionally it does.
I think like by the time that it works in mice, I think it's 3% of the things that work in mice don't fail in clinical trials. So it's like you have an exceptionally narrow funnel that goes from, some kind of molecular screen to some kind of cell screen to some kind of mouse screen to some kind of human screen. And so that pipeline is I would say tens of millions, if not hundreds of millions of dollars to go from, the point where you're like, okay, well, this is a candidate substance to, oh shit, it doesn't work in humans. And that's just money that you've burned, right? And so I always wonder about stuff where it's like terrain theory. If we go back to that, it's this idea that there is some kind of optimal state for the body. There's some kind of optimal pH state. There's some optimal, ionic concentration of salts in the blood and things like that. And I wonder if this isn't also an era of that'll benefit a lot from biohacking, where there's people that spend time on the internet just buying weird proteins and trying them, like
I
peptide
think actually COVID
marketplaces.
was really good for this
Hmm.
in some sense. It's always talked about as such a nightmare because for so many different reasons, but it became really obvious, I think, to people that if you're healthy and strong and for the most part, well nourished and so forth, you saw much better. You had a lot less to worry about, let's say, than people who were
right.
overweight and malnourished, not getting the right vitamins. I think it was kind of this global wake up to actually how much your health outcomes depended on the soil.
It,
That's actually a really interesting point because I feel like there is a really strong contingent of people that still doesn't buy that. Like
yeah,
I think, go ahead.
I think what's interesting about like again kind of like what you're sharing each other is that like someone who's healthy who has a bad reaction probably has like a rare genetic interaction that predisposes them, right? So again, in ALS or Alzheimer's disease, maybe only like 5 to 10% of it is like traceable to genetics. The rest seems sporadic. So yeah, 100%.
And all
I'd
the
be
messaging, right? Like I think people did feel betrayed by a lot of the messaging because a lot of the messaging was more like, you know, everybody needs to get the same medical treatments and the outcomes are independent of anybody's particular
right.
background, right?
And now let's
Just
like dive into this because when you're talking about biohacking, this is super interesting. But what jarred is that people's different genetic backgrounds does exist, right? And so if we take someone, an American like me and compare me to a Sherpa living in the Himalayas, right? They have this genetic adaptation to high altitude climates. And so what do we both need to satisfy? We both need to satisfy this idea of metabolism, of moving a flux of matter through us. And my genetic background is a little bit like I can hang out up there for a while and do some, but it's not like them, right? Their genetic components aren't redesigning what it is to do metabolism. It's slightly tweaking it for a different physical environment, right? So when we're asking what are the things we need to measure in the body, there's a certain few things that we all have to fulfill, and it's getting the right like flow of nutrients through our body. But that changes what we need, changes on your environment. Again, the terrain, right? The biophysics of the terrain change how you'll take up oxygen and want to move that through your body. Right. And so I think biohacking is like one way to look at it, but different groups that are, in a sense, really specialized for certain things, like some groups in Indonesia that like dive deep, right? There's different examples of this. You still have to fulfill these same overarching biochemical goals of your body, but you get there in different ways. And these different ways, there's so much room for genetic detail for all these little ways you can tweak it, but the fundamental problems remain. And so I pose that we focus on these fundamental problems of this mass in metabolism moving through the body and what you need to have that happen, and all of these special adaptations are little tweaks on that. And so as opposed to
Can I know we talked about the Safe Read interview earlier, but I'm curious if you had any thoughts as you said you listened to that interview, and I'm curious if like you had any water revelations that popped out at you, or how congruent his presentation was with what you've seen in your journey.
Yeah, very, very congruent, right? And I think that's what's so powerful to me is, you know, I think that I feel a little bit afraid almost to talk about in scientific circles like the power of diet. But now I'm like convinced by my biochemistry that this has a physical manifestation that it matters, right? There's like a buffering range of your body. And if you're outside that buffering range, this is going to look like disease. And so this is what C3 is talking about, is that you need these particular kinds of chemicals in the correct way and concentration for metabolism to happen correctly. And now we can start to like play with this and abuse this as a cancer treatment, because cancer is a different requirement than your normal cells. So how do you change that in your environment to like encourage them to go back to normal cell behavior? So some things that stuck out to me is, I mean, again, if I listened to his interview 4 years ago, it'd be really different than today. Today, I'm really into it because it makes so much sense to me. I'm struck with his ability to like go through metabolic pathways and highlight individual steps and where this comes about. This is information that I'm missing, but I'm really enamored with, right? Because growing up in biochemistry, metabolism was this detailed class that I like didn't care for much, and now I see the power of like looking into that Roche chart of metabolism, and I feel like C3 offers like, he came into the same way through a totally different path. And I think that there's immense power. So some of the things I learned from him there, like where I would look for other like examples of this, or how I would do that.
Yeah, I've been thinking a lot about how, like, what's the spoonful of sugar to make the medicine go down with what he's saying too, because I find it's a very difficult thing to get people excited about that aren't already.
People have been told that eating meat is bad for them. Like Shiloh has started collecting. I told him that he should start collecting a folder of these. I think that you've begun.
Oh yeah.
Where he constantly comes across studies where it's like scientists discover that red meat is bad for you. And then you look at the data and how they've collected it, and they've combined red meat with processed meats. Or there was another one recently that you mentioned where it was like a study that showed that eggs were really bad for you. And then he went and he looked at the data, and it's like, well, the category that contains eggs also contains like soybean oil. And so it's like, what's actually bad here? Is it the meat? Is it the eggs? Or is it the processed meats and the soybean oil? And that question hasn't been resolved. And so I think that when you talk to people where you're like, look, maintain a ketogenic diet because that's going to fix some dysregulation inside of the metabolic system, they're kind of like, I've been told for years now that eating meat is bad and I shouldn't be eating meat. And so you're fighting against that when you're trying to talk to people.
It occurs to me too that there's a moral ethical angle to this too, right?
Yeah, like, are you going to eat torture meat?
Exactly. So I think a lot of that meat is bad for you is driven by this underlying intuition that the way that we attain meat is really horrendous, which I kind of 100% agree with.
Yeah.
And so asking everybody to stop eating carbohydrates basically moves you back to before agricultural civilization, right? And
I think that it doesn't. Actually, I think it does something worse. I think that it forces you into this incredibly morally uh, horrific animal agriculture model that is the only one that we have right now. Like, I always play this game of like, uh, what is like, who's the Nazi? Where this sounds absurd, but
I
I'm
like that
like,
good too.
okay, so if you
are we the
lived, are
baddies?
we the baddies? But it's like
Yeah.
if you lived in the 1930s in Germany and you were able to look at society, presumably you would be able to point out the ideologies that you were like, these are the ideologies and the things that we're doing that are really, really bad. And we
Mhm.
look through the benefit of hindsight at German society and we're like, how could they have allowed this to happen? Or if you go back even farther and you go back to slavery and you're like, how could they have
I
participated
would
in
never,
I would never have done that. And then you look at animal agriculture and you think about the fact that these are living, breathing animals that are kept inside of cages for their entire lives. They're just abused terribly. And it's hard for me to not feel like that is this one of the biggest moral crises of our time. And so telling everybody to eat keto without having a different way of doing that
That's the
means that
problem.
you're buying into this horrific moral structure where I'm like, that's dark.
Like it really could turn out to be the hand washing of our era. Like
Yeah.
it could turn out to be as
Like
great.
maintaining a proper diet.
Yeah, like avoiding these ultra processed carbs and sugars and stuff could literally, in retrospect, look as insane as people not washing their hands before surgery 100 years from now. But the transition to a world where people can eat high fat, high protein diets without, torturing the hell out of animals it's almost like nobody has a plan for that.
Whoa, I would actually say that the entire story of the last 100 years or so is the story of us facing mass starvation, coming up with tools to escape mass starvation, which is super disease resistant staple crops, which is large scale production of nitrogen fertilizers through the Haber Bosch process, and the distribution of these things across a global system, because the alternative that we were facing down was literally millions and millions of people were going to starve to death. And so I'm like, we
Yeah.
solved that crisis. Like, that was absolutely a
It might
crisis.
have even been the crisis that brought civilization together in the first place.
It might have very well been. And I think even then the next level of that was like the next level of plant breeding. Like there's that guy, I think it was Burbage, Luther Burbank. He was the guy who created a huge number of the fast growing disease resistant cultivars that we use today. So most of the food that we buy in the grocery store today was bred in the last 100 years. Right? Like we started
Right.
the process of domestication 10,000 years ago. But
Yeah.
the foods that we eat today are optimized for growth under these high nutrient, fast yield conditions that are very, very recent. And so I always think about that because I'm like, I don't want to get on the train of like GMOs are bad.
Yeah.
But at the same point, I'm like, I do wonder if by manipulating those crops and not looking to see like what does nutrient content look like? What is the quality of the foods that we're eating? What are the things that we need to put into the soil other than nitrogen? Do we need to replenish minerals? Like what have we lost in the process of making sure that there's enough food that we're not rioting because everybody's starving? Because that's the alternative.
Yeah. I agree. And to kind of like tie this all together, I see this is this thing that humanity has done repeatedly. So when you're asking like who's the Nazi, right? And like am I the Nazi? And you start there. And I like ask myself this question because weirdly I came up with like a couple of things. One,
Mm.
like I have pets. I think pets are kind of strange, honestly. Like I jail these cats around here and we like help each other out. But I'm not sure if like in 100 years we're going to be like, why would you ever do that to an animal? And of course, like our food stocks being an absurd like interpretation of this that's like a lot more poignant. And so I kind of like again like am I the Nazi? Like one of these original things, like humans weren't really maybe like meant to leave the African savannah. If we like go back to evolution, right? Your body's like people like savannas, like it's the temperature, it's the kind of like climate, it's not like super forested, so you can like run, you can survey areas, keep from danger, like everything about it, right? But then we invented clothes and we can go to Antarctica right now. And like a weird consequence of this is that we have like pollution in Antarctica. We have like all these things, right? And so like clothes were this technology that allowed us to like leave where we are at and go to like anywhere, space, right? Clothes lead to space exploration. Space exploration leads to crazy like environmental consequences, good and bad, right? And so what I'm trying to paint at is that like when wondering like, am I the Nazi? Is that there's a high and a low form of every technology that's realized, or every idea. You can like take it in a good way, you can take it in a bad way, and this is like the reflection of humanity's character, right? And
Rudolf
so
Steiner has just entered the chat.
a classic, right? And so yeah, one of the things I like now is again like what dictates where we go with technology, and it seems to like run into this like spiritual thing that we run into. And so again, but the and so in our way there, we also like invented like cities and farming, very cool. Lots of like things that we can do. You can afford to have full-time scientists like me to have these conversations and do this like synergizing because I'm not like collecting this food, but we also have people that are just like siphoning off the top, right? Yeah, a military industrial complex that does a lot of work based off of like our tax dollars, things like this, right? And so there's like a high and a low farm to our tax dollars and these cities organizations, and we're always like trying to figure it out. And we realize that there's a high and low farm to this farming, right? When we leave the hunter gatherer diet, there's all these consequences that immediately show up with farming. And they were going to face the same thing. We invented all these genetic tools to make yield better. It's going to be huge consequences that we're going to have to figure out, that people were figuring out with, well, what went wrong when we went into cities with our health? What went wrong when we started to like do these high yield crops with our health? And frankly, like it's interesting that you mentioned Steiner, because one of Steiner's big things that he wanted to leave with the world was biodynamic gardening, right? As that he, in a sense, foresaw that we were going to struggle with exactly this, with this huge like factory farming operations, and how it would like strip the earth, right, of these like nutrients. And so why one of the reasons why Steiner is so compelling to me, because independently through my like water research, I come to realize how important all this like mass flow and how subtle it is, and how Steiner would describe an unhealthy farm is one where you have to import or export fertilizer. And what fertilizer means to Steiner is cow poop, right? Is what your herd animals like leave, right? They're part of the farm. And so you can't have a farm without the animals to fertilize the soil. And you can measure how healthy your farm is by the imbalance of how much fertilizer you need to add, right? And so he gives these antidotes that, again, kind of like this, for your body. If you don't move nutrients, if you don't have metabolism in the correct way, you get these weird buildups that you need to supplement. And that might be like chemotherapy or other medicines. But there's also a natural way to get that to move through your body as well. And so that's how I think that when we're up against this, it's always the same thing that humanity has been up against. Again, there's a high form and a low form to this, right? Our clothes either give us like, spacesuits or bulletproof vests, right? There's like so many things that come from this. We want to be careful, but we don't want to get rid of these tools, right? We want to take them with us and just hone their utility to us, make them healthy with us.
Oh,
Yeah.
it's like trying to design a future that's more solar punk than it is like a return to feudalism or an Orwellian dystopia, right? Where we're able to take the technologies that we have invented, we're able to do something productive with them, and we're able to pass to some next phase where we are not subjugated by the tools that we have invented and instead are able to actually solve the massive problems of life. Because I think that it kind of is the human project. It's trying to figure out, like, how do you live healthy? How do you live well? How do you take care of the earth? Like, I don't know that the crow or the bison or whatever else is really preoccupied with, stewardship.
Right. And I'm not sure if I want to promote them to that right now, right? That's kind of like our job. I don't want to get rid of it. I want to like master my job first as the steward before I give it to the crows. I think that's also an important thing to remember as well, is that like, you know, uh, I have a single Michael in my head, and I drive this body. And when I was in undergrad and graduate school, I wanted like a lot of Panda Express, but it turns out I probably just was like weirdly influenced by all the microbes in my gut that were used to Panda Express and somehow signaling to me that like I needed this, right? And so weirdly, I'm not just like Michael, I'm like this weird synthesis of many organisms, and they live and die by my decisions, right? And so yeah, whether I like I'm climbing and like scrape some skin off, like sucks to be those skin cells, but it was for the greater good, like my greater good, not their greater good, right? So I myself am driving this really crazy thing where it just feels like me, but I'm hoping that each part of me is doing its own best job. I don't want to manage my liver cells. I don't want to manage my brain cells. I'm hoping that they're doing their thing on their own. And as long as I'm managing, my inside, like everything I need to be, everyone else should like take care of their stuff on their own, right? And so I think this is kind of like a different approach to life in the world, where a lot of our farming, our factory farming, all this stuff, it is because we are afraid of those times before 100 years ago where we would starve all the time, right? And we're trying to like fight against that. And so this is so deep that we try and like be future proof. And in doing that, you make some sacrifices. You start to like harvest a lot more to use for later. But that a lot more you harvest for later is that imbalanced farm that I was talking about with Steiner, right? And so when I'm starting to like make some of these worries or concessions, there's like unforeseen consequences to these micro environments that are also making up my, like if I'm not getting my liver cells the correct nutrients or something, and I start to revolt, and this looks like sickness or cancer, right? Uh, and so I think that uh, what this kind of implies is that you can have this like really top down hyper implemented machine learned perfect society where it delivers everything at your needs, and this is like in a sense to be safe for the future, or I could trust in my cells to do what they do when they need, and kind of like living like more in the moment. And there's like costs both ways, but now I see the cost of trying to like future proof and really plan, have it all like that. I don't want to have to dictate to my liver cell how to live its life. It's way too detail oriented, right? It's not for me. And so I want to do my job. I want to do his job well too. And I think that when we're talking about like the healthy functioning of the body or of societal systems, it does look like this metabolism where you need this appropriate like in and out, and you need everything to like function and be shared, right? Like my excretion of the cow's excretion is the fertilizer for the plants, right? One man's trash is another man's treasure. It's very like just to like focus in on these kinds of things, I think can guide us through these really difficult growing pains that humanity like necessarily must confront, right, for our journey.
I think starvation is an easy one to see the solution for everybody, right? If everybody's starving, it's like, all right, we need to get more food on the plate. But we've reached this point, I think, where people have become very comfortable with the idea that given the present nutrient environment, there's a standard lifespan that people can kind of expect, right? And, that's cool. Everybody's gotten comfortable with the idea that, men live to be 75 or whatever it is, women live to be 80, and like, okay, great. But it's tantalizing to think that punching through that barrier might require a complete reassessment of the entire playing field. Like, what are people dying from? They're dying from heart disease and cancer. What causes heart disease and cancer? Like, it's really crazy. I think one out of every two people is going to experience cancer in their lifetime at this point, right? And should that be the case? Is that necessary? I think we've gotten very comfortable with the fact that, well, if you live long enough, you're just going to get cancer. It's like, well, maybe not actually. Maybe that's only true in an industrial agriculture society. Like, that's really not something that we have to necessarily get comfortable with. And so maybe punching through this next level in terms of health span and lifespan really considers the reevaluation of the fundamentals altogether.
Well, it's crazy, right? Because while you were talking, I looked up if you were to just look at the number of food calories that are produced in the world, we make enough food globally to allocate 3,000 calories a person per day. So, like, we have solved the question of starvation so completely that it's not even
Right.
funny. And now, again, it's the synthesis. We had the problem. It was the fact that people were starving.
And the solution's so obvious for starving, right? With cancer, it's something like
Absolutely.
everything under the sun seems to cause cancer, right? It's like you eat too many hot dogs, you get cancer. You eat too many broccoli, you get cancer. Like, probably just got this video taken off YouTube by saying that.
Yeah.
But yeah, like
It's true about broccoli?
No, I don't. I don't know. Probably there's
That
probably
needs a
some
fact
fertilizer or
check.
like
Oh,
some
yeah.
pesticide or
That's
something.
true.
I mean, my point is that it's very hard to see like a single solution like with starvation for cancer, but I think there might be ultimately.
So that's actually a really good segue, because I wanted to come back to this idea where you're like, okay, so if you look at these amyloid forming diseases like ALS, like Alzheimer's, and you take a CSF sample, the pH is more acidic in the CSF of those that suffer post-mortem. Is this post-mortem or pre-mortem?
This is not post-mortem. This is
Okay.
pre-mortem. So they have an age match control and the patients rather. Yeah.
Okay. So I wanted to get to terrain theory and see if we can come up with something actionable or something that people can try? Like, not necessarily give people medical advice, but like as we're talking about this, I am remembering how back in the day, the first time I lived in San Francisco, I was hanging out with the art school kids. And I was like the only technical person. There was a nurse, but she was more on the side of like patient care. I think I was the only person that was in science. Like I worked in a laboratory every single day. I would go to work. I was like working on tuberculosis, infecting mice with it, doing the horrific things you do inside of a lab. And I remember there was a lot of this kind of holistic, crunchy, crystal health stuff. And I remember there's this one girl that I was talking to who was talking about the fact that she read somewhere that you shouldn't eat sugar because I think her argument was that it alkalinizes your blood. And
Oh,
at the
okay,
time,
interesting.
I was like, she didn't, it's not like she handed me a paper where she was like, look, they've like figured this out. She just like, she heard it somewhere and she was like, is this true? And at the time, I was kind of like, I don't think that that's how bodies work because everything that I understood about biology was that there's like a fairly stiff homeostasis in the body where everything is well buffered and there's not really anything that you can do by virtue of ingesting things that'll significantly change your pH or your electrical state or your osmotic state.
My favorite is the what do they call it? Like pH balanced water or something that they sell for outright
Alkaline waters, yeah, you
alkaline
can buy
water.
like pH, I don't know, like 7.5, pH 8 water.
And I'm like, that goes through your stomach, right?
But yes, it does. However, clearly, if there is some kind of global homeostatic dysregulation of pH that leads to amyloid formation, is it something that can be then modified through behavior or ingestion of something? Or is this just such a big, complicated system that's just gotten dysregulated in a way that is the vector points in one direction only?
Yeah. So a few thoughts on this. First one, I think that's really interesting about like the cancer and alkalization. I just in the chat shared a review I really like by a professor whose work I follow called "The Distinguishing Electrical Properties of Cancer Cells." And there's this one little table where it kind of shows how in normal healthy cells, let's see if I can uh, you have a pH balance of the inside related to the outside of the cell, and that this is flipped in cancerous areas or tumors, right? So this matters. And I also, as you said, I learned also that like the blood is buffered like 40-1 against acid, that your body's so like on these like narrow rails that I think that it gives us a lot of ammunition to like doubt these things mattering. But now and again, like if you engineer a biofuel pathway into an organism, it actually tends not to make that biofuel. It tends to like shunt that energy to where it wants it to go, right? So it always seems like the body kind of gets things to where it wants to go. But it does seem like if you zoom out that this like matters. And what's really cool about where I try to come from on this question is that we could look at that huge, crazy metabolism chart and try and see where each molecule slots in. But it turns out that all those molecules sum to two vectors or two numbers, and it's pH and osmotic pressure. And those two numbers, like everything you put into water, will change those two numbers. And so this gives you an incredible amount of control to like zoom out, and you don't have to worry about the detail nearly as much by just focusing on what is the water potential or the osmotic potential of maybe your blood or a fluid, or in a cancerous tumor, the interstitial fluid versus the intracellular fluid. And so you start to see these relationships, and these are actually what's setting up the driving the mass transfer in metabolism, right? When things are going to flow down or get moved around, it's going to respond to differences in osmotic pressure or differences in pH. So that's why I argue that these two numbers are really important for trying to understand these, because this is actually how we start to get this like actionable answer in a really, really complicated world. Can like kind of everything synthesizes to these two numbers, and everything is going to diffuse to its place of home based on these two numbers. Uh, by what I'm proposing. And I think that's how we can start to like really tackle this, is that, and what's cool about it is it's not this really detailed top down machine learned issue. It's actually cool. You can like measure this. You could take a CSF sample, you can take a blood sample, you could take a sample from a tumor, and you can start to actually like measure this, right? Even that new probe that we talked about, that's like this micro pH probe, you can start to actually non-invasively even kind of make these measurements into a human body, as opposed to biopsy. So I think that what's really hard is that this prompts kind of like a revolution of how we can think about cancer care, screening, and what it means, what to look for. And what's cool is that there's not that many numbers. It's not that detailed. You don't have to sequence the person's tumor. You just have to do a few like relatively simple biochemical assays based on like an extracted sample. And so now what's also cool about this is that there's so many ways to change those two numbers, right? Your diet, your exercise, right? It's kind of like dealer's choice. You can find what works for them. If it just all sums up to that, and all the details emerge kind of like as a consequence of these two numbers, pH and osmotic pressure, then I think that gives us a lot to like go on for feedback to try and hone this in.
Okay, well, so let's say that somebody does a blood test and or they get a spinal tap and they get some kind of readout for the fact that their pH is either too high or too low. One thing that I want to add while I say this is that the when we're talking about like too high or too low, these are like very small changes, right? It's not like you're
Super
going
small.
up to like, from a pH of like 7 to a pH of like 10. These
Yeah.
are like a pH
Point
like
three to point two.
like tiny minute changes. And so I
Right.
have immediately a cut with like several different questions. One, inside of artificial systems, in vitro systems, do these small scale pH changes seem to produce large scale changes inside of osmotic systems or inside of this like liquid liquid phase separation? Because all of this is centered around the
Yes.
fact, like, let me see if I can construct a rough model, which
Yeah.
is that pH is an electrical signal for the conditions inside of the cell or inside of the body, and the electrical conditions change the charge state of the proteins that are in solution, which causes them to interact differently with macromolecules in the environment. And so, if you change your pH and the electrical conditions enough, you break the ability of the cell to properly pull fuel and dump waste. Is that a fair assessment?
Absolutely.
Okay.
Yes.
Okay,
Uh,
so if we go from there, then we have these very, very small changes of pH inside the body. Do those small changes of pH inside the test tube lead to large scale effects for this phase separation that is the structural readout of how water is interacting with stuff? Does that question
so
make sense?
I yeah, there's some stuff for me to like follow up on here, and that is what's really curious is that in the test tube, it's like 99 to 95% water, right? You might have like 1% like proteins, some ions, and buffer molecules in this, but it's hugely water. In your body, it's like 70% water, right? So life takes place. So like in a more biochemically relevant condition in the test tube is one that looks like steak, right? Like totally absurd compared to like that's what's going on. So when small changes in the test tube, uh, they uh you like they lead to like pretty big pH changes, right? And so what's strange is that in the body, to get a cell to do a different behavior, like 0.1 pH percent is like enough, right? Your blood, whether it like absorbs or releases oxygen from hemoglobin, is dependent on a very small like pH change, like point 0.1, right? And so what I'm getting to is that all of the molecules in your body that take away that water, so if there's 70% water, then there's 30% other like molecules, all of those, they have homes for hydrogen to hang out, right? Like think of your DNA backbone. It's phosphate, it's negatively charged. You can put so many hydrogen ions onto that. You can put so many salt molecules onto that, right? So this exceedingly like maximized interface of your body's protein structure gives a crazy big array for these salts and small molecules to bind to, which means that your body can buffer a huge range of stuff. But this also means that like suddenly when you go from the test tube where like a little bit of acid makes this huge change to like a little bit of acid in the body, it is like largely absorbed. But now I'm going to flip it. So now I'm going to start to say like these little changes in the body, this 10% leads to humongous like changes in the body in the cell behavior, because that 10% like that represents way more like hydrogen ions than in the test tube, right? So in the test tube, that number of hydrogen ions might make you like pH 1, but in the body, that number of hydrogen ions changes it like minus zero point 0.1, right? Or something from where you are. And this is a struggle to like figure out. But there is a paper that looked at the hydrogel nature of the cytoplasm. And so inside the cell, there's a cytoplasm. And as opposed to being like a bag of liquid with stuff floating around, it's actually this like really structured environment with all this cytoskeleton. And so you can conceive of it as like a hydrogel as opposed to just a liquid. So this group in 2009 did this. And what they did was they changed the pH around the cells. So they opened up the membrane and they like made holes in the membrane so that the pH would like the acid would get in very easily. And they found that when they deviated from pH 7.2 or 7.4, the cell's volume shrunk immediately if you go up or lower. So what's strange is that you actually this is what happens is that when you start to get out of the normal pH range by like zero point 0.5 in the body, that the cell's material nature, it just loses a ton of water. And this is also because all of those hydrogen ions that were like sucked up by the DNA or the protein, uh, these proteins are no longer repelling each other. They aggregate like in Alzheimer's disease. They get together. And so then they don't need these big hydration shells anymore. So you start to like shrink the cell. And so this is actually really interesting because it also implicates like pressure volume work as a response to changes in the electrical, like the pH changes lead to pressure volume work in the cell as well. But when you're doing these pressure volume work cycles, you need like it's a way smaller measurable pH change. It's like zero point 0.1, but that 0.1 represents way more hydrogen ions than you would get that pH change in the test tube, right? So like 100 the H plus ions in the cell links like zero point 0.1 pH change or something, whereas in the test tube that would make several like go from 7 to 2 or something pH in this example. And this is
Which
just
is
because
for people who don't know biochemistry too well, that's a huge jump. This is a logarithmic scale. That's
yeah,
basically the difference between water and hydrochloric acid.
pretty much, right? Yeah. So because your body has so many sites to accept those acids, your body's like really buffered for this. But then as it accepts those acids and buffers that change, that leads to a change in genetic expression. That's literally like starting to change your body, right?
Okay, so then the next question that I have you come across evidence that macro scale interventions, like the diet that you eat, or the water that you drink, or the supplements that you take, have any obvious effects on pH of the body?
I would say no, but I haven't looked at that too much yet. I think that this is where, as a biochemist, I tend not to look at the body as like a system and zoom out, right? And so, but now because of where my biochemistry has led me, I need to ask these questions. I'm so thirsty. I'm so hungry for like wondering the answers to that. But this is like the frontier, right? And so I tend to think I've built like a biochemical tool or a framework by which we can understand that these things matter. But it's now up to us to figure out and do that like experiment.
It almost seems like the wrong question too, because like the body seems to be this constellation of all these different microenvironments. And so the question is like which compartment is affected? So Seyfried
Yeah.
would talk about some compartment within the mitochondria that's affected by these things.
Sure, but I guess I kind of, and this could be totally wrong, but I'm like, every single cell is bathed in blood and lymph.
It's nice that you bring
And
up lymph. People forget about lymph.
right, lymph is important. Lymph is insane. Like lymph is
Insane.
wild because it's not a duct system in the same way that the capillaries are, where it's like capillaries are maintained. There's like the capillary bed is continuous from arterial and venous vessels. Right? Like you're bringing oxygenated blood, the blood flows through the capillaries, and then it gets carried back through the veins. But lymph just kind of has these ducts that open into the tissues, and then the lymph filters through the tissues, not through the capillary ducts, as far as I understand it.
Right.
And so it's
And
just
I,
literally like bathing the cells, right?
from my understanding, which is rudimentary, but where this is going is what my research then makes me like wonder about is also what Shiloh open up with this like the body as a series of like phase separate compartments, and that's what's cool about the revolution that I've undergone is I no longer think of the body as like membrane separated sacs. I think of them as these like individual liquid phase separated compartments, and they're this interesting interplay between the water and the macromolecules that make them up. And so there's several like versions of this. We have these things called like the nucleolus in your in is a small portion of your nucleus. So the nucleus where your DNA is housed in the cell, there's this thing called the nucleolus, and the nucleolus is this organelle that's not wrapped in a membrane. It's just a liquid phase separated one, and it's where all of the ribosomal RNA is made and processed. And so, of course, like when the cell divides, you're going to shut down your ribosome genesis so you can like package up that area into the chromosome and then segregate, and then you're going to reopen it up. Right. And so you can like dissolve and reform these liquid phase separated compartments depending upon the like what the cell's doing. Now, so there's all these nested versions of this. So the most pressing that I pose is that the nucleus and the cytoplasm actually have the same phase separated relationship, uh, where
As the so like analogy, like nucleus to nucleolus is like nucleuses to cytoplasm.
exactly, yeah. So the nucleus is its own like phase separated liquid environment compared to the cytoplasm, and the and
For people who are listening, right, there's actually a cat climbing on Michael's head just momentarily ago.
Yeah,
That was epic.
just did the whole app things.
So
And so I
I really appreciate that we've managed to keep a straight face through all of the cat antics over the course of the last 2 hours.
yeah, well then I in the living room now, so it's a different game for them. Uh, but I love this idea of like this is how I see the body now. It's all these nested phase separated environments, and the most important one is the relationship between the nucleus to the cytoplasm, because the nucleus and the cytoplasm they obey all the same rules as the liquid phase separated compartments do, including that if you have the same pH inside and outside, you're kind of like the same environment. So during the cell, just its normal state, it has the nucleus is more acidic than the cytoplasm. When you divide, undergo mitosis, you need to dissolve the nucleus, right? So you need to turn the nucleus into the same pH as the cytoplasm. And that's what that microwave lecture that we talked about, that's what that experiment showed, is that as the cell is going through division, they tracked the pH of the nucleus and they found that it changed and went to the same pH as the cytoplasm for cells to undergo division. So that means for a cell to divide, it has to dissolve the nucleus phase separated compartment and then package the DNA and then like reform that in the new cell, right? And so with the same relation as we have the nucleolus to the nucleus to the cytoplasm, there's also the intracellular environment to the extracellular one, or rather the intracellular environment to the blood or to the lymph. And what you start to realize is that the nucleus and the cytoplasm, they're out of equilibrium. And that's how you get so much trafficking, right? The mRNA transcripts need to leave the nucleus and go into the cytoplasm where they're turned into proteins. So, and you're going to need to get the proper nutrients and equipment into the nucleus to replace that leaves, right? So you have this cycle of out of equilibrium between those two environments, and you're going to have the same thing with your cell, where compared to your blood, you need to be out of equilibrium with the blood. You need to have a different phase aqueous compartment in the blood because that's what's going to encourage the appropriate mass transfer from the blood into the cell and then from the cytoplasm into the nucleus. And so it's all these molecules navigating the different phase separated environments that they want to go to. And so blood and lymph are like this other phase separated compartment compared to the intracellular environment, right? And they're going to obey these very similar rules in how they talk to each other and balance.
Have you come across this liquid-liquid phase separation in any other sciences besides biology? Like, is this important in geology or anywhere else?
So there's actually like a 1967, right, like Nobel Prize for polymer science. So people figured out a lot of this for polymer science, and polymer science is like petroleum, like all this stuff. A lot of we use this all over the place, and I think what is missing in my world is that right now it's my role. I think is trying to show cell biologists how this is relevant to what they do. So you can look at like uh signaling of like insulin or some molecule like that, but you can also see how insulin follows an aqueous gradient to get to where it needs to be. So is it signaling? It can be its own like issue as well, or calcium as well. Like when you're firing a neuron, there's like a flash of calcium and sodium. That this is like a signaling event, but there's also like a biophysical like compression wave, and there's an osmotic difference as well to this. So there's like a physical component to all of these signaling events that we're seeing in the body. And I think that's how I add to this system and try and get people to understand in biology how phase transitions show up. But of course, like personally, I receive the world as everything now, right? Like even if you're looking doing like uranium dating or carbon dating, what are you like measuring? You can't measure how old this rock is or how old this like uh, piece of carbon is. What you're measuring is like when it last underwent its last phase transition, right? Like when this rock like went into the center of the earth, solidified, and now it's here, right? So you're almost always measuring like when this thing like last underwent its phase transition. And so now I think that like this model of phase transitions and moving between them is like a really useful lens to approach this world scientifically because it is so general and it shows up all the place now, all the time. And I don't think it's my job to like make everything phase transition in everyone's eyes. I'll do like the cell biology stuff or try my stuff, but I think it does present in other places as well.
I mean, it's really cool stuff. I think that for me, the piece that would be that I would that I want to follow up on after we get done talking is this question of what what can be done from the outside without large scale interventions to change the electrical state of the body through food or the consumption of something. Because I feel like if I'm hesitant to say that this is like a cure all for diseases, but it does seem like there is some kind of, to come back to this idea of terrain, there is some kind of terrain maintenance that's necessary. And I would bet you anything that there is something in the food supply or something in the water supply. Like I always think about the water treatment and the way that almost everybody lives on city water. Like Shiloh and I live on a well, and when I go into town, it's astounding to me how much it smells like bleach.
Yeah,
And I think that when you drink water that's always coming out of the tap, you don't notice it. And like our water's salty as hell, and so it's got its own problems. It's not to say that it's, living on a well has its eternal perks, but it doesn't smell like bleach. And so I
right,
always wonder, I'm like, there's stuff that's in the water that's just people are constantly bathed in, and like, how does that change the pH of the body? How does that change the terrain? Just not
right.
even because you're doing anything, not because you're just eating Twinkies all day long, but just because
Just
like this is the water. Like, oh man, I had a friend that was losing her goddamn mind. Owsley, actually, because she was starting to look into organic farming practices. And she realized that you could label something as organic, but be using industrial wastewater to irrigate the fields.
yep.
And so it's like,
As soon as you list rules, it's all about beating the rules of the game, not like doing the heart of it, right? And so this like shows up in humanity as well all the time. That's why I don't really buy organic because I know that it's like kind of a meaningless term. But like if my friend grew this on their plot or like harvested this cow, I know a lot more about this suddenly, right? And I think that this is like going to be hard to escape. And I think like as you're saying, like this is a terrain problem, and the first thing is you probably don't want to like dump chemical waste onto your terrain, right? Like if you want it to be a healthy ecosystem, like it's got to go somewhere. You got to do something with that. But there's these consequences, and whether it's having like overly salty well water or like too bleached city water. I think what's curious about this approach that I'm proposing is that everyone has a different issue of getting their terrain to match how they want it to be, of getting their environment into the place that they want. Some are too acidic, some are too basic. And you can do even different kinds of like epidemiology, right? What kinds of cancers show up in some region versus another? Is it something that like alkalizes, or does something, is it a small nutrient that like tends to go to the liver or do something else, right? You can totally reframe how you do epidemiology and disease studies by looking at diet and looking at these things because this lens so condenses down what all of those things are doing. If certain types of food or diet have these consequences biochemically in your body, you really do get so much purchase by understanding again, like how do you get to the right terrain you want? And so everything that you do touches it, like matters. And sometimes you're going to need like more base, less base, more salt, less salt. And so I think that we get stuck when like, Steve Jobs' diet, right? Like, is fruit good for you? If you're the average American, yes. If you're on cancer and only eating fruit, no, right? And so again, as I was saying, like there's a good and a bad interpretation of everything. So people tend to like look at the worst interpretation of someone doing diet to beat cancer and give that as like a reason to discard it. But of course, right, like when you start to look into the detail and the minutia, everything always starts to make sense. And that's why I'm posing, right? Why does everything start to make sense? Is because there is this set target that we have, and your stresses push you one way or another, or have complicated interactions. But understanding this like set target and that we should be getting back towards there, I think is like the power here for understanding health science.
And it's like it's really a creepy place to go into because I think there are so many people just trying to make a buck.
Again, and I struggle with this too, because like in my own forays going to like the weird water research, some of the stuff, the more like pseudoscience type topics, like there's so much good biochemistry that's based on that we're just totally missing for like memory of water, that kind of stuff. Like there's really interesting things here. Uh, and so I think that is just like again, I'm sorry.
This is
uh,
amazing. This has been the highlight. I know the water's the
I'm
most
glad.
important part, but the cats really are the stars of the show.
Yeah, he just recently learned to be a shoulder cat, so I think he's got to like do this all over the place now. Yeah, but
I can imagine like a huge industry cropping up. I first floated this idea back when we had is his name Near Barzilai on the
Mhm.
show, the anti-aging guy. But he mentioned he's got a huge clinical practice in New York somewhere, and he mentioned that he'll prescribe exercise and diet to people, but something like 3% of his patients actually take him up on it. And
right,
it occurs to me, like in my own experience, that it's very difficult to adopt diet and exercise unless you find a version of that you're really excited about. And for me, like that was rock climbing, right? Like I
yeah,
wasn't really much into exercising at all before I found climbing, but for some reason, climbing doesn't feel like exercise to me. Like it's
yeah,
so difficult and challenging and cerebral that I forget that I'm exercising, but I actually find myself getting in shape by doing it, which is incredible. And so I wonder if there just isn't like this huge coaching space available if this was going to be commercialized, which seems to be what drives things forward in this
right.
Western world, that people could actually be oriented towards solutions that are compatible with their own interests. And the same thing could be true of diet as well.
Right. And I think that this is historically like shamanism in some sense is like going there and trying to figure out how like things work with someone, or like therapy nowadays so much is like trying to figure out like what works with the individual. And I had the same relationship with climbing. I don't like work out or anything, but I love like problem solving with my mind and the rock wall. So it became my like I not even trying to work out, just like through that. And I think that's so what it is like you love like the lifestyle of climbing, right? And I think that's what we're kind of against. When I earlier we talked about like what's the issue of the adaptation of these ideas, and I posed that like we can show it's effective, but also we need to worry about like society. Society doesn't give people enough time and free space to experiment and try and adapt their life to what does my life look like when I do like wake up and walk every morning. Most people are so stressed with the kids, the school, get like everything right. And so to actually have the time and space for this to be effective of assigning exercise and diet, you need to build a society where people feel that they have the time and energy to do that, right? Like just like the professors around me don't have time and energy to learn to read 80 pages and rethink their life about like how biosciences work. Like I don't blame them. Like no, nothing about their job description gives them the space to do that. It's built so that they can't do that, right? It's not their job in the ecosystem to do that. They're there to get students grants in and like be a different part of the community. It's totally fine, right? So what we're up against is always that we need to like change society a little bit for its like scoring function, and I think that will help.
And in some way, I feel like this is like a biotech adjacent vision or like health tech or something. It feels like it has the capacity to be instrumentalized in a way that is really, really useful for people. Like if the principles hold and there's actually a direct relationship between, pH, osmotic gradients, salt concentrations, nutrient concentrations, macronutrients, micronutrients, that you can use to properly tune the body to a better homeostatic state that's protective against disease, not necessarily curative 100%,
Right.
but at the very least sets you up to have better odds when you encounter the virus that in somebody else might lead to cancer or in somebody else might lead to respiratory arrest. Like, that seems like a no-brainer to me. And I think that it doesn't
Agreed.
follow the same business model as like I can't say that name, as like a large pharmaceutical company that is trying to sell you some kind of blockbuster drug that they've spent billions of dollars of research on. But when we were talking to Seyfried, the thing that I loved is he's like, you can get a glucose monitor, you can get a ketone monitor, you can get the strips, you can figure out what feels good, you can figure out like what foods leave you in the zone, quote unquote. And then you just kind of set it and forget it. And it's something that you can do for yourself, that there's consumer level products out there for it. And this seems like the next level beyond just the emergency of like, you have cancer, we have metabolic
Yeah.
therapy for cancer. This is like, okay, there's an arm of this that's preventative for people that just want to figure out what is their internal state and how to tune it slightly better so that they're less tired or
Yeah,
less
like the biofeedback, as you notice you feel better under certain conditions and so forth, you can hone in on that and correlate it to the numbers, right?
There's this really funny Tumblr meme, which is about this fantasy that people have where they imagine that they go to the doctor and they get tested for a nutrient deficiency, and then they discover that they do have a nutrient deficiency and they begin to take the nutrient, and then everybody is like, you were deficient in the nutrient and yet you prevailed? How extraordinary. And so it's like, I feel like there's this. Well, I
It's
like, I think that there's this desire. Everybody's tired. Everybody's frazzled, spread thin, like too little butter over too much bread.
Mhm.
And there's this fantasy that there's some secret thing that you've been missing. Like,
You just change this one little
if you
thing.
just tweak this one little thing, all
right.
of the sudden you will be elevated to like normal function with everybody else. And it's like,
Yeah.
maybe it's not one nutrient. Maybe like you can't just start taking like vitamin K and turn out okay. But
Right.
maybe if you do tweak 5 or 6 things with some solid scientific basis, or it's evidentiary basis, I don't even want to bring science into it, that you actually can improve your state and you can make your life better simply by making it easier to live. Like, forget about
Right.
curing disease. Like, it's just easier to live because your systems are working. When you eat food, it energizes you as opposed to leaves you like lying on the ground. Like,
Yeah. I think that's a very level way to look at and approach this whole situation, as well as like what the goals should be. And part of what you're saying with like, diet and prevention, it's horrifying to me of thinking of like someone that survived the struggles of cancer, and then they go back and like, do they do the same thing that like led to that? Like, what are all of these things that all these sicknesses are like wake ups to some issue going on, and you can like ideally like treat the disease, but then what do we wake up to is to like what went wrong in the first place. And I think that the other part of this is that, as you like allude to, the body feel like this is a practice. This isn't like one and done. I think that's also this like top down. We desire, how wonderful that it was that one nutrient for you, and you go tell all your friends that changed your life. But like your friends do, it doesn't change their life. And again, like they lead a different life than you do. They may not be as deficient. They have like a different uh bear that they're up against. And I think that we're starting to like realize this part of becoming like a jack of all trades or learning many different paradigms is just opposed to one comes to like flexibility to see that we're all like these unique individuals, and what works for someone doesn't work for other. And so like some science or some onboarding routes or some explanations resonate with some people and miss with others. And I think that this diet, exercise, and health will be the same. And so I think that this also requires a fundamental like different change in how we approach health science, and that it is a practice. Like, doctors aren't lying when they tell you it's a practice. It's not a certainty. It isn't science. It is like, have you tried this? See how it works. See how it feels. It's really important. Like, I think I tended to like discount that when I was younger, but now that's all I believe in, right? And I think that it's also true that like people will delude themselves, right? Like, the placebo is really strong. People want things to work, or they want it to be not diet and exercise and just this one my vitamin that they take up. So they'll like delude themselves into thinking that this works, or I don't know, maybe the placebo actually does cure them. Very interesting that that works there. We should revisit that in the next podcast. But yeah, these little things that work for some and not for others is part of this like practice, and this is just like the going rate of life. And I think that a lot of people get stuck on, it turns into pseudoscience. I had this one or this one crystal worked for me, or this one vitamin worked for me, and so we get these like pseudoscience stories, so people tend to distance themselves from this. And I think that this requires a little bit of change in our societal way of thinking of, again, like experimenting. Experimenting involves being wrong, but our bodies are our own like experimental laboratories, and I think that we should be like a little bit more open to listening to them and figuring out what it's like to play with it. Again, right, like times we shared, like I did crystallography in my youth, and so I wouldn't drink caffeine to handle the crystals under a microscope because that was the alchemy I needed. Someone else didn't need that alchemy, right?
Somebody
So I
else
think
needed
like
to shake really hard. For what they
that's
were
right.
doing.
Yeah, yeah. They really, yeah. The tremor really helps with something. I don't know.
I mean, God, there's just the stuff remaining to talk about is vast. I have like 10 different questions to ask, but I think we should put a pin in it.
Yeah, we got to get you off to this Dino competition.
I would love to go to the Dino competition.
Heck yeah.
But can you
Punch
tell
some
people what
rocks.
dinos are for people who don't know?
Oh yeah. So Dyno stands for dynamic, and so this is a climbing move where if you the opposite of dynamic would be static, and so static is just like moving from one hold to another, being in control the whole time, and dynamic is kind of a leap where you're like leaving the wall, or it's a little bit uncontrolled, right? If you miss your hold, you don't do dynos in the real world because there's a huge cost to failure, but in the gym it's cool
Unless you're
because it's
Chris
just kind
Sharma.
of, oh, that's true too. Yeah, then there's just victory all the way up. Yeah. Uh, no, I think, and so I love that these dyno competitions in the gym where it's kind of this like gymnastic, like let's see, how far these primates can throw themselves up the wall. It's
Yeah, you guys should Google this if you haven't seen a dino competition. It's pretty
yeah,
incredible.
it's somewhere buried is my Ninja Warrior tryout
It really
video
is.
from ages ago.
It's incredible.
All right, man. Well, yeah, good luck with that.
Thank you so much.
Is there a bounty for who wins the dino competition?
Oh, I don't even bother to check anymore. It's usually very small because it's not like a real climbing competition. And so I'm not good enough climber to like do any of those. But things I've won before, like a pull-up bar or something. Which
That's not nothing.
it's not that I still have it. Still works. Yeah.
Oh man. Oh, before you go, you gonna be around May 9th?
So yeah, is this going to be another paradigm drift?
Another paradigm shift. Although we are going to be out in closer. We're going to be on Eastern time, so we'll be
Cool.
a little bit earlier.
Yeah, I would love. I have something all ready to go. I'm
Perfect.
excited. Yeah.
Heck yeah.
All right.
All right, man. I'll email you to follow up.
Yeah.
Same.
All right, Michael.
Thank you so much.
Thank you guys so much for chatting. And again, reading my paper feedback is very helpful. These are really hard problems that we're up against, trying to like figure out what this new science looks like. So thanks for having these conversations with me. I really enjoy talking to you all about it.
Ditto.
Absolutely. Let's do it again.
For sure.
All right. Bye, everybody.
All right. Bye, Michael. Bye, everybody.
See y'all.